All right, this is a little episode on laboratory analysis. We just got our lab results back from some of our '09 wines and an '10 wine. And Vinquery, they fax it over to you. It comes like this. You take the samples to them, and they will run these results. And then we can, based on what we're looking at, we can figure out what we need to do these wines. You can see '09 Cab, the Pinot, the Sonoma Harvest, the Russian River Zin, our '09 Estate. I've also got the Randy Zin '09 Randy down here that we ran a non-filter panel. It talks about certain things here. Certain things I've already found out. Other things we are still what's called pending. So basically looking at all these free SO2s, that's what I wanted to run the big thing on, and they're all looking really good. 20s, 20s, 37 is a little high for our Pinot, but our VA is also a little high too at 0.093. That's as high as we want it, frankly. The Russian River Zin 24. So that means these wines all have enough sulfur in there. Everything is kind of holding in stasis. That, in other words, the amount a little sulfur that I add each month is binding and basically helping prevent the microbes from taking off, spoilage organisms, and also premature oxidation. This Randy Zin, I ran a non-filter panel, so you get the free SO2 there. Then you get over here, you get the malic acid, you get the glucose, fructose, and the VA. Those are the three big numbers that we care about. And if you see, the malic acid is at 4, the glucose, fructose, the residual sugar is at 31. That means this is dry stuff. 0.0, I guess it would be 0.031 grams per liter. That is very dry stuff. And then the VA is at 0.065. This is all really stable stuff. This Randy Zin, it looks very, very promising, and everything's coming along really well. Finally, the Sangiovese, we ran a 2010 Sangiovese, the malic acid. What I wanted to know was this thing done with malolactic fermentation, the secondary fermentation. And you see that 8 milligrams per 100 milliliters. This thing is totally done with malolactic. We're going to now rack it off, take it off the sediment, probably the next day or the day after, and clean out the barrels. And then we're going to put the wine back in and age it, add a little sulfur. Put the wine, what we call, to bed. I'm ready for bed.